Let's talk about cancer. Cancer was first recorded in human literature back around 5,000 years ago in ancient Egypt, but it's been around since much longer than that. Genuine research into the disease didn't begin until the 19th century when scientists labeled it the disease of the cells and began exploring what caused it. Speaking of causes, cancer isn't caused by just one thing. British scientists realized that chimney boys in the early 19th century contracted lung cancer at higher rates than the rest of the population, causing them to conclude that soot was a carcinogen. Smoking causes it too, but it wasn't until the 1950s that cigarettes joined the list. Radiation, processed meats, politics. The list goes on, and it's only going to keep growing. But there's one contributing factor that's garnering a lot of research interest, and college students aren't going to want to hear about it. That's why I'm going to tell you about it. Chronic stress. For about 40 years now, scientists have understood that chronic stress lowers the cytotoxic activity of natural killer cells, an innate immune cell, important in helping the body fight against the growth of tumors amongst other things, which might be one of the reasons why cancer rates are increasing. In this video, we're going to talk about how cortisol, the most abundant endogenous glucocorticosteroid in the human body, is produced and released. I'm hoping by the end that you'll be able to describe how cortisol gets into our cells and activates transcription factors. Not only that, I want you to understand how the cortisol receptors themselves get expressed on the cellular surfaces, because they don't start out there. Then we're going to dive a little deeper into the immunological side of things. We're going to learn the cellular biology behind how cortisol literally suppresses the NK cells and stops them from doing their thing. If you don't know what an NK cell is, that's cool. We'll figure it out. By the end of this section, I want you to see the difference between NK cells that were expressed to cortisol and those that weren't. You should have a general idea of what the natural cytotoxicity receptor is, and by that point, you should be able to explain why normal NK activity is essential to fighting off tumors. Let's get started with the brain. If we dug around deep enough, we'd find an area known as the hypothalamus. This is where cortisol begins its journey as a neuroendocrine signal initiated by the release of corticotropin-releasing factor, aka CRH. CRH binds the CRH receptor on the anterior pituitary gland, which leads to the secretion of adrenocorticotropic hormone, which targets adrenal glands and ultimately stimulates the release of cortisol into the bloodstream. Think of the bloodstream almost like a highway, and the cortisol are like the commuters. To travel through the blood, it needs to be bound to corticosteroid binding globulin, CBG. So I guess that's the car in our analogy. The cortisol CBG complex takes its exit and diffuses into the tissues where it is going to interact with the cells. Now, usually, there's a receptor known as a glucocorticoid receptor on the surface of these cells, but when there hasn't been an initial influx of cortisol, all these receptors are going to be hiding inside the cytoplasm because homeostasis. That's like being the first person to the office in the morning and realizing that you left your keys inside the building. Yikes. Fortunately, some of the cortisol is known as free, meaning that it isn't bound to that CBG, and these can permeate the plasma membrane easy peasy. Once inside the cytoplasm, the cortisol is going to find the GR, which is going to be attached to a multi-protein complex which consists of chaperone proteins, which we've learned about. That includes HSP90, 70, and P23, as well as some immunophilins from the FK506 family. Don't worry about those for now. Once the glucocorticoid binds to the GR, the complex is going to dissociate. The FK506s get activated and they send signals non-genomically through MAPK, PI3K, and the AKT pathways, among other things. Not only that, the GR and its ligand get phosphorylated and it travels towards the nucleus. At this point, it's got two exposed nuclear localization signals that allow it to pass through the nuclear pores rapidly and get inside. At this point, it's going to bind to the glucocorticoid responsive element, GRE, say that 3 times fast, which is a specific DNA sequence that only the activated GR can attach to. This complex regulates the expression of target genes, which are far too numerous for us to talk about here. Just about every cell is influenced by cortisol, and each response is unique in its own regards. But this is where we're going to get specific. What does cortisol regulate inside of natural killer cells? There was a study from 2004 that took place in Tübingen, Germany, where researchers collected blood samples from healthy volunteers. Using a technique known as magnetic cell sorting, MAX for short, they isolated NK cells from the samples by pulling them from the rest of the serum. Some NK cells were given cortisol, some were given prolactin. That's the hormone that makes mammals lactate. while others were given IL-2 and IL-15, and those are stress factors that allow immune cells to grow. And then some were given none at all. Interestingly enough, they found that NK cells exposed to cortisol experienced drastically reduced rates of killing compared to every other cytokine tested, as well as the controls. You can see it in this graph here. Effectors are the NK cells, and their targets were K562 cells, a type of lymphoblast used in immunology research. Obviously, there's a correlation. What's going on here? Let's go back to the nucleus. Remember how we learned that the GR, once bound to the GRE, is going to release a bunch of transcription factors that determine how the cells respond to cortisol? Well, this study measured the quantity of what are known as natural cytotoxicity receptors, aka NCRs. I know, lots and lots of acronyms, bear with me. More specifically, they measured the quantities of NKp46, NKp44, and NKp30 expressed on the NK cells. There's a strong relationship between the presence of these receptors and how effective their NK cells are at killing. They found that cells treated with prolactin, IL-2, or IL-15 exhibited higher amounts of all three receptors. Interestingly enough, 85% of the control cells also express those receptors. Cells that were treated with cortisol, well, you guessed it, were lower on average for all three receptors. Now, these receptors are important for cytotoxicity because they're what bind to the targets and allow them to activate and release all of their deadly granzyme and perforins. When you take away the thing that starts this whole process, nothing happens. It's not like the NK cells go dormant, no. They still replicate as if they're actually doing something. Check out this graph. It shows us that over the course of days, the NK cells the researchers measured were replicating, the cortisol ones included, and in fact were effectively indistinguishable in quantity from those inoculated with prolactin. Therefore, there isn't much of a correlation between quantity and lethality. It all depends on how many of those receptors each of the cells have. Now, this study admitted that it did have limitations. They didn't measure NKG2D. What is that? I don't know. They didn't measure NKG2D, which is another activating receptor that NK cells have. Regardless, it's important to have healthy NK cell functions. They're responsible for killing abnormal-looking cells. Some research has shown a growing connection between NK cell function and autoimmunity. But what's most important is that NK cells are one of the primary cells responsible for eliminating tumors in the body. The study we looked at today was conducted strictly in vivo, but research is finding that people who deal with chronic stress, i.e. cortisol galore, are more and more likely to develop cancer, and the suppression of NK cell function seems to be one of the likely culprits. Alright, that was a lot of stuff. Let's recap. Cortisol is made in the brain and secreted into the bloodstream where it travels all around the body. Eventually it enters the tissues where it can interact with the cells. If they don't have receptors already on their surfaces, the free-floating corticoids permeate into the cells where they bind to the intrinsic receptors in the cytosol. Then from there, kinases get activated that cause non-genetic signaling. The receptor enters the nucleus where it behaves as a transcription factor by binding to GRE. This can do several things, including causing the expression of glucocorticoid receptors on the surface of cells. In NK cells, this causes the downregulation of NKp46, NKp44, and NKp30, all of which are natural cytotoxicity receptors present on natural killer cells. Without these, NK cells can't form their connections with the targets and consequently, they can't kill anything. While NK cells are important in stopping tumors from spreading in the body, so when their killing is augmented, this can lead to the increased likelihood of cancer developing in the individual. Cool. Well, that's all. Here are some questions that I think you should think about. Alright, here's the first question. Where does the glucocorticoid receptor, aka GR, once bound to cortisol in the cytoplasm, go? Pause the video and think about it. It becomes phosphorylated, and then it travels to the nucleus to function as a transcription factor. Alright, next question. What is the name of the DNA sequence that glucocorticoids regulate? This is known as the glucocorticoid responsive element, also known as the GRE. Alright, last question. What are the three natural cytotoxicity receptors, NCRs, that are essential for NK cell cytotoxicity. That would be NKp46, NKp44, and NKp30. Thanks for watching.